Hello and welcome, dear students. How are you all? So, I welcome you all to another session of biology, and I guess you must be all ready because you must have seen the by now the results of all the NEET and the AIMS examinations. Okay, so maybe you are following up on all the questions. Okay, so let's get ready. Let's get started. Okay, so please also mark your attendance so that I will be able to follow it. All right. Centers, please report your attendance. Okay. Yeah, I believe the students are doing it right now. Okay. So 7 students present. Okay. All right. So before we start now, we'll do a little bit of quick revision of what we did in the last class, in the previous classes. Okay. So, all right. Still more are coming. All right. By the time they come, I'll be doing a bit of revision. So you will be able to follow all that. Okay. All right. So, dear students, in the last class we have done about introduction of the 5 kingdom classification, and we have done a little bit of the primitive kingdoms. Okay, so I will be talking about kingdom Monera again for some obvious reasons that you know that questions related to gram staining, questions related to differences between gram positive, gram negative bacteria. Reasons asked that why archaebacteria, why the archaebacteria have extreme adaptations. What is the reason for their extreme adaptations? So questions like that, and the particular name of cell which fixes nitrogen in cyanobacteria. So these type of questions are asked from Monera. These are the most obvious things that you must know besides the other things. Okay, so I always try to focus on the most important things which are repetitively asked and which are interesting to know, of course. So I try to put those things first along with other subject matters. Okay. All right. So Churachandpur, you are telling me you have a new student joining today. Okay. So I welcome you, dear student. Please feel welcome. And feel free to ask any doubts that you have. So you will have to complete a bit of notes. Okay. All right. Okay, so Churachandpur, I believe your name is Anna. Welcome, Anna. So I will tell you how to catch up. Okay, so let me tell you about the whole syllabus. Okay, so the class 11-12 syllabus of the NEET examination, if I give a brief introduction how it is categorized, so please try to follow me up. Okay, so the basic thing in biology is the living organisms, the biological organisms. So other students can also follow up who might have just want to revise it. So biological organisms have some basic properties. So we call these the properties common to all living organisms. Okay. So these are growth, metabolism, reproduction, consciousness. Out of these properties, growth and reproduction are such properties that they cannot be constituted as properties which are characteristic of living organisms only. For example, growth is present in non-living and reproduction is present in living, but it has some exceptions. Some people cannot reproduce and most people can reproduce. Okay, so things like that happen. Otherwise, metabolism and consciousness, they are the properties common to all the living organisms in the world. Okay, not only that, if we go to Mars and find some alien biological matter, suppose somebody goes to Mars and finds a mass of amoeba-like tissue, and he touches it and it recoils back, attacks that person, astronaut, maybe an Indian astronaut. So we will call it that this matter is living, okay, because it is showing consciousness, it is showing a stimulus, and it has metabolism, it is breathing or something like that. So these were the properties I taught to the students in the first class. That is where I start. Now after we study about the properties of living organism, next thing becomes how many living organisms are there in the world. Okay, so do you know how many living organisms are there in the world, Anna? Okay, there are 1.7 to 1.8 million living organisms in the whole world, and you can understand that so many species and new species are being discovered every year. All right, new species are being discovered every year. 1.7 to 1.8 million living organisms. 1.2 million out of these are animals and 0.5 million are plants. So we cannot learn the name of each and every organism. Is that clear to you, right? So what do we do? Whenever the number of anything we need to learn becomes too huge, too large, we always try to categorize those all the living organisms into categories, and then our job becomes to learn about those categories. Okay, for example, you might not know everybody that's in your coaching. You might know everybody that's in your class, but you do not know how many students are there in the coaching overall, okay, or in the school overall. You don't remember them by their names, but you always know what category that person belongs to. Suppose he's a very religious person, you will ultimately know from somebody that I do not know his or her name, but he or she is a religious person. Or he or she is something like they throw a lot of parties. So you will know them by their categories, okay? So we like to assign people some categories. We like to call them names, nicknames, so many things, okay? So we can easily remember nicknames better than we remember names. Similar is the case with classification. We try to learn the categories. Suppose you see a spider-like organism crawling somewhere. It has 8 legs. You will call it a spider. You do not know the exact name. You do not know the exact detailed classification of that spider, but you will be able to identify it with a spider. Now you go in the, you are walking by the road and something is coiling and moving in the grass, and it's long and slender. So you will call it a snake by default. Okay, so organisms might not be known to you exactly, they might not be identified with you by you in great detail, but you always try to identify them by certain categories. That is what classification is all about. Okay, classification is all about maintaining learning categories, and we are going to learn a lot of categories here. That's what I am here to teach you. Now after categorization, classification, we learn about different types of classification. There is 5 kingdom system of classification: natural, artificial, phylogenetic. This is something about classification. Okay, let's leave it up to that. I'll talk it in detail later on. Now after classification, our job becomes to study about these categories in great detail, and there are 5 categories, 5 greatest categories. The greatest category is kingdom. So there are 5 kingdoms: kingdom Monera, bacteria that is, Protista, fungi, plants, animals. Kingdom Monera, Protista, fungi. These 3 are studied in great detail only by the BSc students. BSc in microbiology or any other stream, they study in BSc about these 3 kingdoms. Our job at right now is to study in some little detail about plant kingdom and animal kingdom. Other kingdoms, we learn only a short introduction. Monera, Protista, fungi, you will only learn hardly 1 to 2 pages about them. That's all information you will require. But plants and animals are the whole 11th and 12th syllabus. Now let's see how. Plant and animal kingdom will be studied in great detail. You will learn all the categories, the subcategories, okay? So you will learn some subcategories which are below kingdom. Okay, so there are 7 categories out of kingdom, which is the greatest, but then there are lower categories, lower subdivision. We will learn these subdivisions, and after that, plant kingdom and animal kingdom. Now, we will have to study some more details about it. This is what is in your NEET syllabus. In class 11th, after studying plant and animal kingdom, then you study how we look them, suppose somebody, like you are a new student, and you are going somewhere, and you suddenly enter a class. So everybody will see you, okay, they will greet you with a smile, and they will try to judge you partially by your appearance, okay. Suppose you are wearing specs. So you might have seen this, if somebody walks in with glasses on, we try to form a mental image that this person, she or he, must be a studious person, okay. They must be studious person, that's why they are wearing those specs. It's like sort of things which goes in our homes, in our friends circle, that we call that he has got some specs on because he is studying a lot in the night or sometime, okay. That's why his eyes are strained, and that's why he is specs. We know that scientifically it is incorrect, but we try to say it, okay. So first of all, you will try to judge anybody by their external appearance, okay. So that is what we do in plant kingdom, animal kingdom. We try to look at them from the outside. So we see that plants, how they are looking from the outside. It is called morphology. It is called morphological study. So first we will study morphology of plants, then we will study anatomy of plants. Anatomy means detailed inside knowledge, okay. Inside knowledge is called anatomy. Just like and you will talk to somebody and you will come to know how they are actually from the inside. So that is called anatomy. We will do the anatomy. Anatomy is studied by studying cross section, vertical section, longitudinal section. That is, two types of sections are there. So anatomy of plant, done. After that, there is morphology of animals, most animals, and then the anatomy of animals, okay. Then comes, your job becomes to go on the detail side. Then you will study the differences between cell, plant cell, animal cell, differences. And then you will study in the next chapter, after cell, there is the chapter called cell division. How do plant and animal cells divide? What are the types of cell division? Now this is another unit. After that, your job becomes to study how the cells of plants, they are working in synchronization with each other, how they are helping each other. So this is called the physiology of plants. Photosynthesis, respiration, you will remember these things, right? Photosynthesis, respiration, mineral nutrition, lots of chapters are there. Plant physiology. After that, animals, okay, we are studying everything in a very systematic way, sequential way. So after that, you will study human physiology or animal physiology. All right. So that is your human physiology, animal physiology. All right. So human physiology done, how animal cells are working with each other. This is your class 11th syllabus that we will cover here. After that, we will cover the 12th syllabus, because NEET paper comes from both 11th and 12th syllabus. So 12th syllabus is starting from reproductive system, and reproductive system, first of plants, then of animals, okay. Reproduction is a whole unit. Then we will study the inside theory, the inside information, how plant and animal cells transmit their information from one generation to the other. That is genetics. Then you will study how genetics, genetics, how we manipulate genes. That is called, manipulation of gene technique is called biotechnology. This is in your class 12 syllabus. Then we will study about kinds of diseases, manipulation of genes, biotechnological applications, so many things like green revolution, etc. Then the last unit in class 12 syllabus and your NEET syllabus is ecology. How plants and animals, we have studied them separately. Now how they are working with each other and maintaining the balance of the ecosystem along with the inorganic environment. That is ecology. So this is a complete syllabus that we are going to cover here of biology, okay. And I am going to take botany, and Meenakshi ma'am is going to take zoology. So Anna, I gave you a short introduction what the syllabus is all about. I hope you can understand how your this is a very structured, very disciplined way that I can tell you about. So NCERT goes by this sequence, and I also follow this sequence only. So I have started from the beginning, and I will sequentially in a stepwise way move forward. So I have covered in the last class, students, please assist your colleague, your classmate. So please tell her about the notes, please give her the notes also about the things that have been covered. And so you will cover those notes, and after that you can share your doubts with me. You can ask them on WhatsApp group or ask me in the class, no problem. Okay. And then there will be assignments also, and I'll give you a few PowerPoints at the end of each chapter. So the living world chapter is too short. There is nothing so detailed in giving that. But after 5 kingdom classification system is finished, that is after plant kingdom is finished from my side, then I'll give you a PowerPoint presentation also that how all these, like it will contain a mini summary of all the notes, okay, that are all that I have taught you, they will contain that. But your job is to cover the notes that I have taught in the class, because the notes are very detailed, and PowerPoint presentation has a lot of images, maybe some videos also, and that the purpose of that is to give you a brief summary, and you can cover the most important things and learn things in a more better manner. Okay, so please try to catch up by the next class that we have here. All right. So dear students, I believe now everybody has come. Okay. So students, please can you give me somebody can give me a summary of the last class? Students, please please come forward. Can anybody give me a brief summary of the last class, what topics that were taught, without looking at your notebook? Without looking at your notebook, can you remember what I was teaching in the last class? At least give it a try. If you cannot remember, then I will assure you that, yeah, you can open your notebooks. But first of all, whenever I ask anything from you, try to remember just without looking at your notebooks, okay? I will also give you a memory technique today. I want to teach you something which will help you remember things faster and better, okay? So first of all, yeah, the topics that were covered. All right. So first kingdom Monera we were covering, all right. And in Monera, look, kingdom Monera is very huge. It has Archaebacteria, it has Eubacteria, it has Mycoplasma, it has Rickettsia, it has Chlamydia. Okay. So we were doing types of Archaebacteria, all right. Great. Now, do you also remember how many types of Archaebacteria we had discussed based on their habitat? That was the criteria. Okay. Yeah. Look, histone protein is a protein which actually it is the protein which is present also in eukaryotes. Okay. Very good. You remember about it. Okay. I'm happy that you remember. Yeah. Very good. Okay. All right. So I believe you can sit now. Yeah. Please. Yeah. That will be fine. Okay. Let the others also answer. Very good. Okay. All right. So let me give you a brief introduction. I'm not sure you people remember, because there is a gap, and whenever there is a large gap, students, you will always have this problem that you will try to forget things. So please try to, like, I want to tell you about something. It's a very interesting technique. So how you will cover it? Okay. So look, I want to teach you about something about neuro-linguistic programming. Okay. Neuro-linguistic programming. Look, this is a scientific study on how to memorize better and how to learn faster. All right. So, two things I want to tell you. First of all, try to remember this graph. So this graph has two coordinates, two axes. First is memory retention on the y-axis, and on this is their time period. It is a graph plotted between time period and memory retention. Okay. So when I try to draw this graph, I'm taking a time period of 1 week, 2 weeks, 3 weeks, 4 weeks. Okay. So scientists have found a correlation between how much revisions you should do and how frequently you should do a revision in order to remember things. Look, memory is of two types. Memory is of two types. First type of memory is called short-term memory. Okay. Suppose you have an exam tomorrow, and what happens is, because of this exam, you are studying the day before, and you suddenly learn some things, and you instantly go and give the exam. This is called short-term memory. Now, short-term memory is variable in people. Some people have a very good short-term memory. They can prepare for the exam just overnight. Okay. Most of the students have this ability. Okay. Short-term memory. But in the NEET examination or any examination which focuses on a large syllabus which is covered over 2 years. Now, for this kind of examination, you need a memory which is called long-term memory. And short-term memory is converted into long-term memory only and only if you try to revise the things. Please do not think that a 2-year syllabus you can instantly prepare in 1 month or 1 week. It is very difficult. It is extremely difficult, and not everybody will be able to do it. But anyway, what we are trying to do here, we can definitely cover a 2-year syllabus in 1 year. That can be done, but that requires the help of teachers and your constant practice. Okay. So try to remember this. Now you have the more so reason is because now you are not going to school. So you will have a lot of time. So if you try to do your syllabus, you can easily finish it within 9 to 10 months. That is the standard procedure. Now scientists have found that the relation between memory retention and time period is that memory is retained, short-term memory, the first graph that I am plotting you, 100%. Okay. So nobody remembers 100%. Okay, so you have studied something, so the graph, the maximum you might be able to remember is about 95%, below 100%. So I'm not drawing it above that. So without revision, so somebody is not revising. So this graph will continue to fall, memory retention. The area under this graph, it represents how much you have actually remembered. This area of the graph represents how much you have actually remembered. So if anybody is not revising within 1 week, okay, so you have learned something today, but you do not revise it on Sunday or Saturday. So you will constantly lose the important points, and ultimately that topic will be lost to you. You will have to start it over again. That will be very problematic. But if somebody is doing a proper revision, all right, if somebody is doing a proper revision, then what will happen? He will remember, somebody is learning something, so he will remember about, so he is doing a weekly revision, okay, and if he is doing a weekly revision, the graph will be something like 95%, 93%, 92%, he will remember about 90%, okay. Still 100% is very difficult, okay. You cannot remember everything. So please always try to remember everybody has a limit, okay. Do not try to push yourself too much, but do not overestimate yourself and do not underestimate yourself. Okay, this is important. So you will remember about 90 to 95%. Okay, and if you are doing a very good revision, you might be able to remember 100% of the things, but that requires utmost skill. Okay, but I'll try to consider what most people are here. Okay, so I'm drawing this graph for most people. Now, if somebody is revising after 1 month, okay, he will remember about 85 to 90%. This graph is for 1 week, 3 weeks, or 1 month revision. Okay, 1 week, 1 month revision cycle. So this is also good. Okay, you cannot revise everything every week. So instead of that, at least at the end of the month, you will revise everything. If you can do that, you will remember about 85% or 80%. Okay, maybe in some cases 90%. But let's be realistic, you will remember only about 80%. And who is doing revision? Weekly revision, okay? He is also covering the revision that he did in the past week, okay? So these kinds of people are the toppers. These are the toppers, okay? 90% retention, weekly revision. This is only possible when somebody is revising weekly, okay? So please choose your category, what kind of person you are, and if you want to raise your standard, you raise your type of effort, so please try to remember these facts, and I hope you will be able to do better. Now one more thing. Now I'm going to tell you how you will use these another technique to help you remember faster. Look, I'm drawing a face of a person. Okay. All right. So there is a purpose of what I'm trying to show you here. If you want, there are three things here. This is a actual eye movement. It is an eye movement pattern for memory. All right. So what I wanted to show you here is something like this. AR, KR. Okay. KR, AR, VR. Now, suppose somebody is sitting in front of you, and you are asking the your friend or whoever is sitting in front of you a question, okay? And he tries to remember something. So, do you remember in which direction your eyes move? You will instantly try to move your eyes towards the roof, okay? In right direction or in the left direction. I am trying to tell you here, if this graph has been found true for 80% of the people, when anybody is asked something, okay, when anybody is asked something, they try to remember and recall those memories by looking in right hand direction on the upper side, okay? They try to remember those things. Sorry, there is a change here. VR dash, AR dash, KR dash. They try to remember those things while looking in upward direction. You find this true or not? Whenever a teacher asks you something, you start looking towards the roof, right? Okay? So this is one thing. Now you are, suppose anybody asks you that, what is the name of that song you were listening to? You are trying to remember those lyrics. So you close your eyes and you move your eyes towards the left or right direction. Okay? This is one thing. Now somebody asks you that, have you eaten that piece of cake, those pastries or those cakes or something? So you will try to close your eyes, or you will try to look towards the floor, and you will lower your gaze, lower your eye movement, lower your eye direction, and you will start looking towards the floor in right side or left side direction. When we try to remember anything based on past experience, you try to move about 80% people move their eyes in right direction. Visually remembered, auditory remembered, and kinesthetically remembered. These are three things. But if you want to learn something better, faster, in order to be able to learn some things, if you want to remember a diagram, you should, 80% of the people should close their eyes, or if they want to, do not close your eyes, open your eyes, but move your eyes in the opposite direction, in the left hand direction on the upper side, okay? So that is called VR dash. AR dash, you are a person who tries to learn everything by repeating it, by saying it aloud. Okay, there are so many people that they will go open the copy and they will start moving on the chair, and they will try to remember things. Okay, so if you want to remember it even faster, try to move your eyes in between towards the right hand side or the left hand side. So you will be able to remember it better if you are a person who learns by the sound. Okay. Now, if you are a person who writes everything down again and again, you use a rough copy and you practice it again and again, again and again. So you will try to, if you want to learn it faster by solving things, by solving questions, by writing those things down, try to move your eyes in the left hand direction in the lower side. Now you will have to analyze yourself, and you will see that about 80% people, if they want to recall something, they will look in right hand direction. And the other is for learning it. The right hand side, the graph is for recalling it. Okay, so one part is recalling, one part is learning. The other people will have the exact reverse. Okay, so 20% people try to learn things by looking in the left hand direction, and they want to they want to recall something, they will look in left hand direction instead of right hand. And if they want to learn something, they will look in right hand direction. Scientists have found that if you show a particular type of eye movement, okay, in trying to recall something or in trying to remember something, if you follow this pattern, you will be able to increase your performance by up to 30%. Minimum, okay. A minimum increase of 30% has been seen in the individuals who are able to follow this graph. Okay. So just wanted to tell you about these things. Try to follow these principles. You can also search for it on the internet. It is not something that I made by myself, okay. It's a scientific study. It is called Neuro-Linguistic Programming, NLP. So you can also search for it on the internet about the detailed how memory retention works. So this was a technique to be able to help you better, okay. So I hope you do something better about it, okay. Now let's try to revise, okay. Let's see what direction you are moving your eyes in, okay. So in the last class we had covered archaebacteria. Archaebacteria. Now archaebacteria are a type of bacteria which are the most ancient organisms on earth. They are the most ancient organisms on earth. Alright. Archaea means ancient. Now we have categorized these bacteria into three categories: methanogen, halophile, and extremophile or thermoacidophile. Based on the type of habitat the archaebacteria are found in. So archaebacteria are of three kinds. What are those three kinds? First is methanogens. Second category is halophiles. Third category is thermoacidophiles. Alright. So methanogens are found in anaerobic environment, mostly in the rumen of cattle. And they produce methane also, and they are able to convert cellulose into glucose. They are able to convert cell wall material into glucose. That is why cows, buffaloes, they eat a lot of grass, because they can obtain glucose from it, but we cannot. Okay, so they do this with the help of methanogens. Then come the halophiles. Halophiles are those bacteria which are found in a very extreme environment called salt water, seawater. Seawater. Seawater has about 35% or 33% concentration of sodium chloride and other salts. In this highly saline environment, salty environment, very few bacteria can survive, and such bacteria, a very good example is halophiles. Okay. Now the property of halophiles is, if you take them out of seawater and put them in fresh water, they will die. They cannot tolerate salt concentration below 10%. Okay. So 10 to 30% or 35% bacteria, 10 to 35% salt concentration, they will grow very well. Okay, alright. I also told you that there is a bacteria which is a very important halophile, and it is used in the clearing of oil spills. Does anybody remember the name of that bacteria? I told you about a bacteria which is a halophile, and it is very important in ecology. In clearing of oil spills, whenever mineral oil, petroleum oil a ship is going from one place to one country to other, from Gulf countries to India, and suppose in the middle there is a storm and the ship is overturned and the oil is leaking over the ocean surface. Okay, so do you remember I told you about a bacteria which is able to decompose the mineral oil? Do you remember? Yeah. Can you just give me the nickname of the bacteria or maybe the full name of the bacteria? Yeah, very good. Pseudomonas putida. This is called a superbug bacteria. It is used in the clearing of oil spills. Very important. Okay. All right. Third category is thermoacidophiles. Thermoacidophiles are those bacteria which are able to survive in hot acidic lake, 80 degree to 100 degree Celsius boiling water, and in that water high concentration of sulfuric acid is present. Sulfuric acid is a very, very strong acid. Okay, sulfuric acid is an extremely strong acid, and if you drop a bone into it, bone will be dissolved. If you drop a piece of skin, it will be dissolved after some time. Okay, it's a very strong oxidizing agent, a very strong substance. So sulfuric acid lake, in this environment, some bacteria are able to survive. These are called thermoacidophiles. Now, archaebacteria, they are the most ancient bacteria. Okay, so suppose there is a like anybody, we can say, like you might have seen in newspapers that there are people who are over 120 years old, 110 years old. Now, whoever is a very old person, they will have a huge family. All right. Similarly, archaebacteria is so ancient, it is about millions of years old, billions of years old. So we can say that all organisms, including humans, eukaryotes, everything, if you believe in evolution, according to scientists, it's not my theory, okay? So according to that, if it is so ancient, then everything must be a descendant or a children of archaebacteria. And this has been found to be scientifically true. Everything, including us, we bear some similarity to the archaebacteria. Okay, so that is what I told you in the last class, that archaebacteria have some similarities with prokaryote, other prokaryote, that is eubacteria. They have some similarities with eukaryotes, us, and they have some unique features. Okay, unique feature is that they have a unique kind of cell membrane, monolayer. Similarity with prokaryote is they have a unorganized nucleus, no cell organelle is present except ribosome, and they have a very primitive structure, cell structure, very primitive cell structure. But the unique, similarity with eukaryotes is that they show presence of histone proteins. As somebody very good, in a very good manner pointed out, they have histone protein. And I told you, I had to explain you in the last class what histone protein is. So what is histone protein? Histone protein is a protein block, and we coil 2 turns of DNA on top of it. Actually, it is 1 3/4 turns, but for simplicity's sake, try to remember that about 2 coils of DNA are coiled like spring on one histone protein. Then another 2 turns on another histone block. In this way, DNA is packed in a very well systematic fashion inside a nucleus in eukaryotes. But exceptionally in archaebacteria, histone-like proteins are found there, which are eukaryotic character. So this means that eukaryotes are descended from, or they have an ancestor in archaebacteria. All right. So this was the information that is very important for your exams. First, you need to remember similarity with eukaryotes, similarity with other bacteria, and unique feature. Then you have to remember the 3 categories of archaebacteria based on the type of location or habitat where they are found. Okay. So let me ask you a general question. There is a sewage plant, okay, which is processing sewage. I'll ask you a quick question. A sewage processing plant, a sewage processing plant uses anaerobic and aerobic bacteria to decompose waste materials. ठीक है? All right. Now my question to you is, the question starts now. A person entered the anaerobic waste chamber and became unconscious. The possible reason for this is, look, this was actually asked as a value-based question, okay? But I'll give you the options. I've changed the options now, okay? I'm just giving the options, otherwise in the CBSE examination, options were not given. So some very interesting questions are there, and this is one type of that question. Methanogen, harmful bacteria. Third reason is viruses. Fourth reason is fungal infection. So anybody can guess the correct reason why a person might have become unconscious? I need an answer. It's not very easy also. In a way, if you think it is easy, it should be fairly easy for some people. I have already told you in the last class, whoever is attended might remember the hint that I gave you in the last class. I told you some such type of incidents keep happening. Somebody has entered an old well, old building or tunnel, old secret tunnel, and they might have become unconscious. The reason is always the same. There is a secretion of something. I have not received any answer from you people. I believe some people are walking up to the camera. I just need the option. Nobody has answered. Is there a chat problem? Okay, I can see you raising your hands. All right, okay. All right, so whoever thinks first option is the answer, raise your hands. Whoever thinks first option is the answer, raise your hands. Okay. Whoever thinks second option is the answer, please raise your hands. Okay. Third option, anybody? All right. Fourth option? Okay, I believe everybody is clear on that. Yeah. Answer is methanogens. So please be very clear. Methanogen, methane gas is a poisonous gas. Okay. Not a very toxic poisonous gas, but if inhaled in large amounts, a person will become unconscious. Okay. So anaerobic waste chamber produces large amount of methane and along with some other gases, but methane is the main component because archaebacteria are the only bacteria that will be able to do this job. So we use in a gobar gas plant, in sewage processing plant, and so many other processes like industrial waste processing plant. We always use methanogen in anaerobic chamber. First we use aerobic digestion, then we use anaerobic digestion chamber, and finally the waste is very much decomposed. it is nearly equal to clean water or clean waste. Okay, so the answer is methanogen. All right, good. Very happy that you are able to do it. Anyway, so let's continue. So we have studied that there are 3 types of bacteria, archaebacteria. Now let us study about eubacteria. Look, I will try to make you people remember. In kingdom Monera, there are archaebacteria, there are eubacteria, there are mycoplasma or pleuropneumonia-like organism, then there is rickettsia, and then there is chlamydia. These are the things we have to cover. So archaebacteria is over now. Let's move on to the eubacteria. All right. So anybody can tell me what is the habitat and habit of eubacteria? What is the habitat and habit of eubacteria? Do you remember? All right. Eubacteria are cosmopolitan. Cosmopolitan is a term which means everywhere. On your hands, on your hair, on your table, in air, water, soil, above soil, many kilometers above in the atmosphere, many meters below the soil. Eubacteria are present everywhere. Okay, they are present everywhere, so they are called cosmopolitan. Habit means nutrition. Nutrition wise, they are very much diverse. Some bacteria may be autotrophic, they can make their own food, but most bacteria are heterotrophic. That is, they obtain their food by feeding on living organism or dead organism or behaving as parasites. Okay, so they may be saprozoic, holozoic. Okay, they show this kind of behavior or habit. So we will write about habitat and habit. First of all, habitat and habit of eubacteria. Okay, first of all, EU means true. EU means true. So bacteria, true bacteria. Okay. They are found everywhere. Cosmopolitan. All right. So they are found everywhere. They are cosmopolitan, and the interesting thing is they can feed on a large variety of things. They can feed on your hair, they can feed on your nails, they can feed on our dead tissues of the skin. They can also invade organisms from the inside. That is, they can become pathogenic. They can enter inside and harm us. Everybody is producing vitamins because of which bacteria? Can anybody answer me this? Suppose you have a bacterial infection. Okay, like I am probably having a staphylococcus infection. Okay, that is why I am coughing. I am having a staphylococcus infection, which is responsible for common cold. Okay, so my diagnosis is staphylococcus bacteria. So I am having to take an antibiotic. But anyway, please tell me, what is the name of that bacteria which is living in your intestine and it is helping you? It is producing some vitamins which are very essential for you. Does anybody know the name of that bacteria which is inside you? Everybody has it. Okay. The name of that bacteria is E. coli. E. coli is a bacteria which lives in your colon. Okay, Escherichia coli, Escherichia coli or Escherichia coli. E. coli is the short name. It lives in the colon, and it is responsible for converting undigested food into vitamins, and particularly vitamin B12, vitamin K, and vitamin B complex. The most common way of saying is vitamin B1, B2, B6, B12. But we can simply say vitamin B complex. That is why if you are suffering from an infection and antibiotics are given to you, the doctor also prescribes multivitamin tablets, because if you are being given antibiotics, bacterial population, they will die in your intestine. Now you will have to suffer a deficiency of vitamins. That is why whenever you go to a doctor and he gives antibiotics, if he is a good doctor, he will also write some multivitamin tablets for you or multivitamin syrup for you. That is the reason. Okay. Anyway, so they are found everywhere. Every place that you can think of, bacteria are found everywhere. And what do they feed on? They may be feeding on any kind of material. They can decompose almost any kind of material. So you will say they are nutritionally diverse and can be autotrophic, but autotrophic bacteria are very few. Try to remember, very few. And most of them are heterotrophic. Heterotrophic may be decomposer or parasitic. Alright, so this is about it. Now students, do you remember, you must have drawn a diagram of a bacterial cell. Do you remember the structure of bacterial cell? Yes? Okay, what you will do is, let us draw a bacteria. How a bacteria looks like, what are the spatial features. So I will draw a general structure of bacterial cell. A general structure of bacterial cell. All right. The thing on the outside, this is the cell wall, and then on the inside I will have to draw a cell membrane. This is the cell membrane that I am drawing here. Okay. Now there is the genetic material or the nucleoid. I was thinking of giving this diagram to you as a homework, but then I thought that I must explain to you right now, otherwise you will have a lot of confusion. Now there is another thing here. You will draw some dot-like structures. These are the ribosome. These are the ribosome. Anyway, and this is the flagellum. Okay. They might be sometimes small granules called phosphate granules. So I am drawing that also too here. Also, there might be an infolding of plasma membrane. It is called mesosome. So let us start labeling all these things now. This is the cell wall. This is the cell membrane or plasma membrane, right? Now this is the ribosome. Okay. This structure is the flagellum. Okay. This is the nucleoid or the genophore. There are many names for it. I will write all these names here. Genophore or primitive nucleus, incipient nucleus, okay. These are phosphate granules, cytoplasmic inclusion. Now this structure here this is the mesosome. Okay. So labeling is complete. This is the general structure of bacterial cell, but one thing is remaining. There is another structure called fimbriae and pilae. Sorry, they do not grow on the inside. They're only present on the outside. This is the fimbriae. Main purpose of fimbriae is for attachment. And then there is another thing called pilae. Pilae serve only one purpose. They are for conjugation. They help in sexual reproduction in bacteria. Pilae. Now this is the complete structure of bacterial cell. Structure of bacterial cell. So please finish it, and as soon as you finish, I will move on. Okay, you have done this. Everybody has done it. Should I move on? I'm not able to see clearly whether you have finished or not. Look, there are a lot of details here. I'll have to discuss those details also. Look, I am using many colors. If you are not able to, look, everybody does not carry colored ball pens. So you can, if you have at least two, black and blue, that will work good. Otherwise, if you want to switch between colors, and you have only one kind of pen, you can use a dotted arrangement, okay, draw dots in some places, so that you might understand the difference here. Okay. So I have used a characteristic feature like Fimbria and Pili, they are almost of the same height, but Pili are somewhat shorter, Fimbria are larger, longer. So there are small differences. So please try to draw it accurately, because that will be important. Nucleoid is the primitive nucleus, unpacked nucleus. It has many names. Attached to the cell membrane by the help of mesosome. Okay. All right. So I'll move because I believe everybody has done it. Okay. If somebody has not done it, please raise your hands if somebody is still unfinished. Everybody has done it, right? So if anybody is still drawing, let me know. I can see you. Just raise your hands. Okay, everybody has done it. Great. Few people draw fast. Very happy. So now let's try to write the names of these things. First of all, the thing is cell wall. Okay. Cell wall is made up of a substance called murine. What is the composition of cell wall? You can say cell wall is made up of murine or mucopeptide. Okay, and this murine or mucopeptide is made up of two substances, NAG and NAM. NAG stands for N-acetyl glucosamine and NAM stands for N-acetyl muramic acid. If you want to know, I'll write these names for you. N-acetyl glucosamine, NAM is N-acetyl muramic acid. These two polymers are cross-linked with each other. They are cross-linked with each other, and that forms the complete structure of bacterial cell wall. Okay, and interesting thing is the cross-linked cell walls, they are broken down by an enzyme which is found in our saliva, tears, all body secretions, and that enzyme is lysozyme. Lysozyme can break down the cell walls of bacteria. Okay. Anyway, also cell wall synthesis is inhibited by penicillin antibiotic. Okay. So there are some antibiotics which inhibit development of cell wall, and without cell wall, most bacteria, they will die. So cell wall inhibiting antibiotics, they are considered broad spectrum antibiotics. They are usually very successful in treating diseases. So we have used them for a long amount of time. Dr. Alexander Fleming was the person who discovered penicillin, but I hope you already know. Anyway, flagellum. Actually, flagellum is considered a cell organelle, but thing is there are some differences. So flagella is not present in every bacteria. So cell wall is done. Another unique thing is cell membrane is just like every other cell membrane, so we do not need to write about that. Flagella, we do not need to write about any special thing about flagella. Then we need to write about something called mesosome. Very important. Mesosome, questions have been asked. What is mesosome? So try to imagine what is actually mesosome. Mesosome is an infolding of plasma membrane. Mesosome is an infolding of plasma membrane. This is an infolding. So we'll write mesosome is actually an unfolding of plasma membrane. Mesosome is actually an unfolding of the plasma membrane. What is the function of mesosome? It is considered to be the precursor of bacterial, not bacterial. It is considered to be the precursor of eukaryotic mitochondria and chloroplast. It is considered to be precursor of eukaryotic mitochondria, because they are also having membrane-like structures on the inside. Mitochondria and chloroplast. Okay. What is the function? It contains respiratory enzymes, so it is believed to help in respiration. Function of mesosome is asked. So what you will write is, mesosome is found to contain function. Respiratory enzymes okay, it is found to contain respiratory enzymes and helps in respiration. Because it contains respiratory enzymes, so it is believed to be helping in respiration. That is a function that you need to remember. Now let us write something about the nucleoid. Lot of questions, lot of queries are based on nucleoid. What is nucleoid? It has a lot of names. Nucleoid has a lot of names. Also known by the name of genophore or by the name of bacterial chromosome. Single chromosome actually. Incipient nucleus or primitive nucleus. Incipient nucleus, primitive nucleus, genophore, bacterial chromosome, all these are names of nucleoid. Okay. And how does it look like? It looks like a particular complete mess. Like you take a lot of thread and you fold it like this and that and this and that. Okay. So you try to jumble it, or just try to take out your headphones from your pocket, and those headphones will look just like nucleoid. Okay. So randomly, it's a random mass of fibers. Actually not fiber, it is a random mass of DNA. Actually, nucleoid contains double-stranded circular covalently closed DNA. So let me write about it. What is nucleoid? It is actually a partially organized, some people call it partially organized, some people call it unorganized, okay, partially organized single double strand of DNA. Look, this is a bit confusing. I am writing single also, I am writing double stranded also. Actually, DNA has a double helical structure. Okay, so double stranded or double helical, they mean the same thing. But there is only one chromosome in the bacteria. Partially organized single double stranded single comma double stranded DNA molecule arranged with the help of non-histone proteins. Okay. Now please remember, there is no membrane outside the nucleoid. I will also write this. This is the difference, main difference between nucleus and nucleoid. There is no membrane outside, no cell membrane, no plasma membrane outside nucleoid. There is no membrane outside nucleoid. Also, what does it contain? It has DS-CCC DNA. Double-stranded. It's a short form. It will help you remember. Circular. Covalently closed DNA. Okay, so this is a full form. Okay, so this is enough for that. Plus non-histone protein. Plus non-histone protein. Also, there is a thing called plasmid. What enzyme does the mesosome contain? Look, if I tell you the real thing, somebody has asked me, Shillong, what enzyme does the mesosome contain? Actually, cytochromes are contained, but if I tell you the most accurate present information, according to that information, mesosome does not actually exist. But in NCERT it is written, so it is written. But very recently, 1 or 2 years ago, it has been concluded that mesosome is not an actual structure. It is actually a result of cell damage. Okay? Whenever plasma membrane is damaged during microscopy, it becomes mesosome. But you do not need to remember these things. You will only remember that plasma membrane becomes infolded like a banana leaf, and that forms the mesosome. So we will have to remember this also. We will have to wait until the NCERT changes its information. So right now, we will remember this only. Now there is another thing. You will find a lot of questions on this thing. It is called plasmid. Now if you see the diagram of if you see the diagram of the bacterial cell, now the diagram of bacterial cell, you can see here, we cannot see it very clearly. Here the thing which is present, it is called the plasmid. Yeah. here, like this. So plasmids are present here. These are extra chromosomal segments. You can draw this here. These are plasmid. Okay? These are plasmid. Now you can just draw a small loop there. Okay? You can just draw a small loop there. Alright. Plasmids are extra chromosomal segments of DNA. Extra chromosomal. Actually, this definition needs to be learned. That's why I have to draw this. Also in class 12th also, you will find that in biotechnology, like PBr 3222 or PBr 322. E. coli has a plasmid. You can see the diagram if anybody remembers. Plasmids are extra chromosomal segments of DNA in bacteria. Alright. So they are like small loops. It is also double stranded. Okay. Now this DNA is also double stranded, circular, covalently closed. Plasmid DNA is also double stranded, circular, covalently closed. You can just write the short form. You can just write the short form. Alright. So whatever I am drawing here, you will find questions. Plasmid you will find questions. Cell wall you will find questions. And nucleoid you will find questions. Lot of questions. So this is very important for your NEET examination. So this has to be covered. Okay. So this is done now. Now there are fimbriae, pillae, function of fimbriae, function of fimbriae. These are hair-like structures. You just need to write a single line only here. These are hair-like structures. On the surface of bacterium. Okay. And help in attachment. Okay. This is done. Pillae do not help in attachment. They help in conjugation. Just need to write this only. Actually, two pillae of two bacteria come together. They form a structure called as conjugation tube. We'll cover this later. Okay. Fimbriae done, pillae done. You can also write about the cell, like cell membrane is not very important. Cell membrane is same everywhere. Flagellum, not much has to be written. I will actually discuss flagella types and flagella absent. This has to be done. Now we can come into bacterial classification on the basis of types of flagella, bacterial classification on the basis of type of shape. Bacterial classification based upon shape and size. Basically, it's on basis of shape. Size is not important, so you can write it in brackets. It's understood. If the bacteria is spherical in shape, if the bacteria is spherical in shape, it is called coccus. Coccus is a name given to a bacteria which is spherical in shape. Now, if the bacteria occurs singularly, if the bacteria it occurs singularly, then it is called monococcus. Monococcus. Single. If two bacteria are there, okay, sorry. We finished that. All right. Okay, if we finish that, then there is no problem. I believe we have also done a bacterial classification based upon flagellum. Flagellum we have covered, right? Atrichus, cephalotrichus, the monotrichus, peritrichus, amphitrichus. Okay, all right. So, bacterial classification based upon gram staining also then we must have done. Try to remember that. Gram staining. Have I covered gram staining or not? Okay. Okay, great. All right. So I only need to tell you about the last part. All right. So now you know gram positive, gram negative. They also have a difference in their cell wall. Like gram positive cell wall has almost no lipid content, but gram negative bacteria has more lipids in their cell wall. Also, gram positive bacteria, they are less pathogenic. Gram negative bacteria, they are more pathogenic. Gram positive bacteria contains, in their cell wall, they contain teichoic acid. It is absent in gram negative bacteria. So I asked you to write some differences between gram positive and gram negative bacteria. Okay. All right. So I'll give you an assignment. I believe that will be better. So we'll do that cover in that only. Okay. So gram positive, gram negative bacteria differences and all that, and a lot of things we'll do in that. Okay, great. So eubacteria you have done. Cyanobacteria is also part of eubacteria. I believe we have also done that. Okay. Cyanobacteria. Cyanobacteria are called the blue-green algae. And you must remember there is a very important question that is asked from this part, that there is a thick-walled cell present in cyanobacteria. What is it called? You remember the name of this cell? Heterocyst. Okay, just trying to make you remember. Heterocyst. Okay, so this has been covered. Cyanobacteria play an important role in nitrogen fixation in the soil. Nitrogen is fixed, they make the soil more acidic. Okay, so these things happen. All right. Okay, so now we have to do the other types of bacteria, and they have a very important place in the Guinness Book of World Records. It is called mycoplasma. Okay. Economic importance of bacteria, I'll give you a short list. Okay, I'll give you in the summary. Okay, economic importance of bacteria, like bacteria produce a lot of antibiotics. They are important in a lot of biological activities, harmful activities, useful activities, diseases also. We'll do that later. Now, mycoplasma is a part of kingdom Monera. It is something separate. Okay. Mycoplasma, it is also called PPLO, called pleuropneumonia-like organisms. Pleuropneumonia-like organisms. Okay. Pleuropneumonia-like organisms. Mycoplasma were discovered by two scientists. Their name were Nocard and Raux. Nocard and Raux discovered mycoplasma from pleural fluid of cattle. Pleural. Do you understand what pleural fluid is? What is pleura? Anyway, I'll explain. Don't worry. Suffering from pleuropneumonia. Therefore, they were given the name PPLO. Okay. All right. Okay, so let's come up to this. The cattle suffering from pleuropneumonia. Now, what is pleuropneumonia? It's a disease, a type of pneumonia occurring in the pleural membrane. Look, our lungs, the lungs that we have, they have a membrane out the outside. Lungs are covered by a membrane called the pleural membrane. And pleural membrane, inside of the pleural membrane, there is a detergent-like substance. This detergent-like substance, it prevents friction between the membrane and the lungs. Okay? Lungs are protected by an outer membrane called the pleural membrane, and in the pleural membrane there is a fluid which prevents any kind of friction developing between the lungs moving and the pleural membrane. It's filled inside the membrane. Now, this pleural fluid, if it becomes less in quantity, a person will face huge problem in breathing, because whenever he will try to breathe, the fluid is very less, he will face a large amount of friction, inflammation in the lungs just by breathing. He is not hit by anything, he is not hit by a truck, he is not hit by a car, but every time he tries to breathe some air and he tries to take a deep breath, it will be like getting hit by a car or a truck on the lungs. There will be inflammation, friction, so breathing becomes very difficult, and it becomes very short breaths. Short breaths, a person cannot do anything. Like pleuropneumonia, it's actually a disease of cattle, but pneumonia is a general disease occurring in the lungs. All right. So any organism suffering from pleuropneumonia, they will face this kind of problem. So they isolated mycoplasma and found that it was even smaller than the normal sizes of bacteria. Later it was found that mycoplasma is the smallest known living cell. That is why I'm saying that it can find its place in the Guinness Book of World Records, because it's the smallest known living cell. Other things that you need to remember, and that they are asked about mycoplasma. All right. So PPLO or mycoplasma do not possess cell wall. Do not possess cell wall. Okay, they have a cell membrane, and the interesting thing is their cell membrane is triple layered. 3 layers of cell membrane are found. Also, 3 layers of cell membrane are found. One more interesting thing, if they do not possess a cell wall, what will that imply? It will imply they do not possess a cell wall, they only have cell membrane. That means that they cannot be eliminated, they cannot be destroyed where any antibiotic which inhibits cell wall formation. We can inhibit the growth of other bacteria by giving them an antibiotic which inhibits the growth of cell wall, the formation of cell wall. But we cannot treat mycoplasma infection just by giving penicillin-like drug which inhibits cell wall formation. Alright, penicillin is ineffective against mycoplasma. You can write penicillin is ineffective. No effect of penicillin, because they do not have a cell wall. So they already know how to survive without a cell wall. A very interesting thing about mycoplasma is they cannot survive in any medium, they cannot grow in any medium without eating a lot of fat. Okay, they love fat. Actually, they love steroids, they love cholesterol. Mycoplasma require steroids. Steroids, I do not mean just testosterone. They will love testosterone also. That's a type of steroid. But the thing is, all steroids have a cyclopentanoperhydrophenanthrene nucleus. No need to remember this long name. I just wanted to tell you that there is a kind of nucleus. It has a long name. If that thing is present in any substance, we will call it a steroid. Mycoplasma requires steroids or cholesterol for growth. They cannot be grown on any synthetic medium without cholesterol. They cannot be grown on any synthetic medium containing nutrients without adding cholesterol. Generally, the mycoplasma are obligate parasites. Obligate parasites means those organisms which will try to infect a particular kind of host only. Okay, but they can grow on other things also. So we do not try to confuse ourselves in that. Anyway, mycoplasma is the smallest known living cell, and the record of even the smallest cell among mycoplasma goes to Mycoplasma gallisepticum or Mycoplasma genitalium, and this bacteria is found in sterile males. Yes, this Mycoplasma is responsible for sterility of males, and it was found, incidentally it was discovered. So it is quite possible that sterile males, they might be suffering from Mycoplasma infection, and in such a case they can be treated by giving them antibiotic dosage. Mycoplasma gallisepticum or alternative name of this bacteria, of this mycoplasma, is Mycoplasma genitalium. Is the smallest known living cell in the all of the kingdoms. Okay, all kingdoms is the smallest known living cell. Found in urinogenital tract sterile males with infection. So, diseases has been asked. Mycoplasma gallisepticum causes which disease? So, answer is sterility in human males. Sterility in human males caused by mycoplasma. Question has been asked. Another, look, mycoplasma is famous. Most common questions from mycoplasma are types of diseases. So there are three diseases you will remember. First disease I already told you about. Second disease which is asked is bunchy top of papaya. Sorry, bunchy top of papaya. Bunchy top of papaya caused by mycoplasma. Also, witch's broom disease in plants. Plant becomes like a broom shape, a witch's broom, okay, like a Harry Potter's broom. So plant becomes of all the branches, they become almost fused together, and not exactly fused also, they became separated on top. So fused at bottom, separated on top, it gives a broom-like appearance. So branches and leaves curl inwards. So this is called the witch's broom disease. Also leaf curl disease, three diseases you will remember. In plants, mycoplasma causes. In plants, mycoplasma causes bunchy top of papaya, second is witch's broom disease, third is leaf curl disease. Sometimes leaf curl diseases is in papaya, sometimes leaf curl disease is in brinjal, eggplant, but this will be sufficient. Okay, so mycoplasma related all questions have been done. All right. So mycoplasma is over. That's all we need to know about it. Now let's give an heading, separate heading, rickettsia. We do not need to write great detail about rickettsia. Let me tell you about rickettsia. Rickettsia is a disease caused by rickettsia organism. It causes yellow fever or Rocky Mountain fever. There was a doctor by the name of Ricketts. He went to study this disease. He was bitten by a mosquito and he became infected by rickettsia and he died there. But his research became important before his death. So this organism was named in the honor of that doctor, Edward Ricketts. So it is called rickettsia. It was discovered by Ricketts, who was studying Rocky Mountain fever or yellow fever. All right. After his death, the organism was named after his name only. So Rocky Mountain fever, yellow fever is caused by rickettsia. That's all you need to know about rickettsia. We do not need to study about it in great detail. Next organism is chlamydia. Chlamydia. Chlamydia is an organism, okay, so it's called intracellular energy parasite or ATP parasite. That's all you need to know about it. It is an intracellular energy parasite. It goes inside the cell and it begins to eat the ATP only. It needs the ATP parasite. Yeah, I was going to give you the break. Just a second. I understand you need a break. It's okay. Now rickettsia is done, chlamydia is done. With this, kingdom Monera is over. Okay? Kingdom Monera is now over. Now you can have your break. Okay? So after your break, I will start kingdom Protista. So you can have a 5-10 minute break. Okay? So kingdom Monera is done. So I was waiting for you this kingdom to get over. So now after this break, we will start Protista. So please, you can have your break. Okay? So I hope you all are back. All right. So now we have to start kingdom Protista. Okay. We settle down quickly. Look, we do not have much class time left, so do not expect I can give you a longer break. Okay. So please settle down quickly. Kingdom Protista. Kingdom Protista. Okay. Now, kingdom Protista, these are the unicellular eukaryotic organisms. Unicellular eukaryotic organisms. Okay. Up till now, what we have studied, these were the unicellular prokaryotic organisms. Now, the organisms that are there, they are the unicellular eukaryotic organisms. All right. Unicellular eukaryotic organisms. It includes the unicellular eukaryotic organisms. Also, there is a problem with this kingdom. This kingdom includes organisms that might have been kept in kingdom fungi. This kingdom includes organisms that might have been kept in kingdom Animalia, kingdom Plantae. So you can say the boundaries of this kingdom are not well defined at all. Okay, say the boundaries of this kingdom are not well defined. Sorry. The boundaries of this kingdom are not well defined. Also, this is the most nutritionally diverse kingdom. Organisms are phototrophic, heterotrophic, heterotrophic, and they can be a mixture of both. That is called mixotrophic. So this is the most nutritionally diverse kingdom in the entire five kingdoms. You can say most nutritionally diverse kingdom. Look, kingdom Monera has also diversity, but this has even more diversity than kingdom Monera. So that is why I am saying most nutritionally diverse kingdom. Is Protista. Why? Because organisms may be photoautotrophic, heterotrophic, and mixotrophic as well. Photoautotrophic, heterotrophic, and maybe mixotrophic as well. Okay. Now, what are the organisms included in kingdom Protista? Organisms are diatoms, dinoflagellate, euglenoids, slime molds, and protozoans. Okay. What are the organisms included here? We'll have to study them one by one. Sorry. Yeah. It includes Diatoms are also called chrysophytes. Chrysophytes included in diatoms. Dinoflagellates. Euglenoids. Slime molds and protozoans. Okay, these are the organisms that we have to study. These are the organisms that we have to study. Does anybody have a question? If you have it, you can ask me. This might be a little confusing for some people. Look, these are unicellular eukaryotic. So we can include unicellular eukaryotic organisms which can resemble animals. We can include unicellular organisms which can resemble fungi. For example, slime mold. Protozoans resemble animals. Still we are keeping it here. Dinoflagellates and chrysophytes, they are basically like plants only. They are like alga. So they might have been included in kingdom Plantae, but because they are unicellular, so we are keeping them in kingdom Protista. Euglenoids are a unique type of organisms which can produce its own food in presence of sunlight, but when food is unavailable, it can become heterotrophic. It starts eating other bacteria, other alga, other organisms. So it is called mixotrophic. These are the organisms we have to study. So first of all, put the heading chrysophytes. Or chrysophyta. Okay. Chrysophytes. It includes, chrysophytes includes, it includes golden algae and diatoms. Look, golden algae are called golden algae because they resemble, like they are slightly yellow brown in color, so they can be called golden. It includes golden algae. Another name of golden algae is desmids. But please remember, desmids are very less in number, and the main population of the main population of chrysophytes is diatoms and diatoms. Okay. Diatoms, the most common question related to diatoms is, first is their cell wall, so related to cell wall, silica, uses of cell wall of diatoms. Next question is, what organisms are the most responsible for photosynthesis in oceans? The answer is diatoms. Okay. So things like that are asked, so you can write here. Diatoms are the most important photosynthetic producers in oceans. I'll tell you the statistics. About 55% of all photosynthesis in the world is done in oceans. 55%. And out of the 55%, 90% photosynthesis is done by diatoms. 90% of 55%, that comes around 45% or 46%. Okay. So that is why they are the most important photosynthetic producers in oceans. They are a part of plankton. So whenever you have been to a riverside, lakeside, you might have seen some green floating mass. That is called plankton. Okay? Plankton represents the floating plants found on the surface of water. So you can write they float passively in water currents. Okay? They float passively in water currents. Alright? So if they are floating passively, that means that they do not have any locomotory organelle. Okay? Called plankton, and locomotory organelles are absent. Locomotory organelles are absent, but locomotion still occurs. It's not even in your NCERT, I'll tell you about it. But locomotion occurs by mucilage propulsion. It will be a bit difficult to understand how this works. Diatom has a soapbox structure. It secretes a small amount of mucilage. It comes out from the back, absorbs water, suddenly expands in size, and pushes the diatom forward. So this is a complicated physics phenomena. So I would not like to discuss this here because it's not asked and it never will be asked. That how mucilage propulsion works, explain the physics of forces in mucilage propulsion. So just remember this, that will be sufficient. Alright. Diatoms have a soapbox-like structure. What is a property of soapbox? Soapbox means one half is bigger, one half is smaller, and the bigger half covers the smaller half. Diatoms have soapbox-like structure. Diatoms have soapbox-like structure. Their outer shell or cell wall is composed of two halves. Fitting one on top of another. Okay, so two halves fitting one on top of another. This is called soapbox-like structure. One is larger. Okay, let me draw this in a different manner. Let me draw upper one bigger. This lower one shorter. This is the structure of all kinds of diatoms. They may be of very different shapes. They may be circular, triangular, rectangular, polygonal, but always they have two halves. Okay. Also, their cell wall is encrusted with silica particles. Silica particles. Silica, chemical composition of silica, anybody knows? Chemical composition of silica. SiO2, silicon dioxide. Can you find this silica anywhere? Anybody is wearing, some girl is wearing those artificial jewelry earrings or something? Artificial jewelry earrings or your watch. There are some quartz pieces on it. Some shining white colored crystals which are not made of glass. Okay, so artificial jewelry contains a lot of those tiny things. So for you, you can also find in them in your mother's artificial jewelry and a lot of things. A lot of earrings contain those things. Okay, that's not diamond, that's quartz. And quartz chemical composition is silicon dioxide, SiO2. That is the same thing which is present in sand, the shiny particles in sand. That is silica particles, and that is also what is present in the case of diatoms. Okay, and when light falls on these small tiny particles, it is given out in every direction. It is called total internal reflection, TIR. Alright, and what is the advantage? It does not matter at which angle is the light falling. It will be thrown into each and every direction. So it becomes very important in the case of glow sign boards. I'll have to explain and expand this point. After the death of diatoms, after their death, first of all, they were plankton, they were floating. After their death, they sink down to the ocean bottom. Why were they floating before? Because they are so heavy, they will drop down. Why were they floating? They were floating because they had gas vacuole. Air bubbles were filled inside their body, the cell. So that is why they were floating. But after death, no new bubbles will be produced, no gas vacuole will be produced. So they will become heavy, they will drop down. After their death, they fall down on ocean bottom and after millions of years, okay, after millions of years, they will be deposited one on top of another. One on top of another. This becomes a large diatom deposit. It is called diatomaceous earth, and it is very much used in our daily life and forms huge deposits over thousands of years and millions of years. Huge deposits over thousands of years, millions of years. These deposits are called diatomaceous earth or diatomite or Kizil Guha. Now Kizil Guha or diatomite is related to the Nobel Prize. Okay, it actually is related to all the Nobel Prizes over 100 of years. Nobel Prizes were started in the year 1900 and now it's about 119 years. So diatomaceous earth is responsible for all the Nobel Prizes given over 119 years. You will ask me, how is that possible? Somebody is asking me, what is mucilage? Okay, you want to see mucilage. The best way to see mucilage is go to your home, go to the local market, the vegetable market, and purchase some lady's finger. Okay, somebody is asking me, sir, what is mucilage? Okay, Senapati, purchase some lady finger and cut it. And when you cut it, try to touch the cut portion of lady's finger. When you touch it and apply it in your hands, you will notice a mucilaginous substance. That is mucilage. Okay? Any substance which absorbs water and suddenly becomes expanded, it is called a mucilage. Aloe vera, it contains mucilaginous gel. Alright. Coming back, I told you diatomite is responsible for Nobel Prizes. Why? Alfred Nobel discovered dynamite. The thing is, making dynamite, before dynamite, there was no explosive available which could blast rocks away easily. Low intensity explosives were present, like gunpowder, and they were very unsafe. You press them together, they will explode, but that was very unpredictable. Lots of accidents happened. So everybody was looking for a safe explosive and a powerful explosive to make bridges, to make roads on hilly mountain areas and so that. So the problem was there was no safe explosive, there were only unsafe explosives, and one of those explosive was trinitroglycerin. Okay. So trinitroglycerin is very easy to make. You mix nitric acid with glycerin. Very easily available things. Glycerin is available easily. You mix it with nitric acid and heat it. After triple nitration, glycerol will be converted into trinitroglycerin. So this is a very, very powerful explosive. If one drop of nitroglycerin, I take it and drop it on the floor, it will explode. It is very sensitive to shock. So it was very unsafe explosive. What Nobel did was, Alfred Nobel mixed diatomite with nitroglycerin, and it became a very, very safe explosive called dynamite. Okay. So nitroglycerin was so unstable that if a truck carrying nitroglycerin was transported from one place to another and suddenly there was a roadblock or something, going on top of it, the truck will explode, blowing everything in sight. The whole bridges, nothing will remain. But dynamite is a very safe explosive. It will explode only when it is ignited with the help of a fuse. Otherwise, it will not explode. So Alfred Nobel made dynamite, made a lot of money. Everybody needed dynamite, and when he died, he had a will. He did not leave the fortune to his family, something like they were separate. He had no family actually, no real sons. There was something. He was more of a scientist and more occupied with himself. Anyway, so what did he do? He said that after my death, all my property should be transferred to a bank, and whatever interest, okay, bank will pay some interest to me. So whatever interest is accumulating from that, take that interest, divide it into 4 parts: physics, chemistry, maths, biology, and and economics. Physics, chemistry, biology, and economics. And all these 4 fields, they will be given a Nobel Prize from my side. Okay, divide it into 4 parts and give each part to scientists who have done amazing things over 1 year or many years. So every year, some scientists will be given their prizes based on their hard works. So we try to choose only one winner, but it's not possible. So there may be 1 winners, 2 winners, 3 winners, 4 or 5 winners also. In case of biology, it is possible. Chemistry, it is possible. Whenever same research field is there, many scientists are there, and each has given an equal contribution. So they might be given Nobel Prize, all of 4 or 5 of them. So this is the story of diatomite. Now, where do you see dynamite? Sorry, diatomite. You have seen traffic police boards, okay, glow sign boards. So whenever light falls on it, even it's like a very much dim light or anything, it will shine equally on all sides, and we can easily see that, yeah, the signal is shining there, the limit of road must be there, we should not go off road or beyond that sign. Okay, so it becomes a warning sign. So road blocks and so many things contain that, and railings, road railings are painted with night visible paint or glow sign paint. It is made with the help of diatomite particles. Okay, anybody who is very much like into K-pop culture, you can see the girls and I think even the boys use something. They use something like a glitter. They use it on their cheeks and some places. So that glitter is coming from diatomite. That glitter is coming from diatomite. Okay, so it has immense uses, lots and lots of uses. Cosmetics, lipsticks, so many applications, they contain diatomite. All right. Also in industry, we use diatomite for clearing oils and syrups. Like oil and syrup, filtration is very difficult. They filtering these substances and to remove impurities is very difficult. But diatomite is a very fine powder which lets oils pass easily. It is also not destroyed easily. Like if you use any other thing, it will ultimately be destroyed. Oil has such an action, but diatomite is very resistant, so it is very useful in filtering oils and syrups and these things. So we'll write these things. Diatomite is having huge applications used in filtering of oils and syrups making dynamite making night visible paint glow sign boards, etc. All right. Also, if diatomaceous earth deposited is found somewhere, there is a huge chance that petroleum might be discovered nearby. So, like, I'll keep it simple. It's having huge cosmetics also. Okay. So this is about diatoms. Now do you want me to start the other thing called dinoflagellates? The other thing is dinoflagellates. Look, it will be, I think it will be a bit difficult. I'll leave it here. Okay, I'll just tell you what are dinoflagellates. Look, dinoflagellates are unicellular photosynthetic protists having two flagella. Also, their cell wall is highly coated with cell plates. Cell plates. Therefore, they are also called armored protists. These cell plates are made up of cellulose and pectin. There are two flagella. This one is called the transverse flagellum. This one is called longitudinal flagellum. Each flagellum rests in a groove on the cell wall. Each flagellum is placed inside a hole or a groove called the transverse groove and the longitudinal groove. Transverse groove and this is the longitudinal fissure or longitudinal groove. Now, what are these cell plates? These are the cell plates. These are made of cellulose and pectin. This is the general structure of dinoflagellate. Now, I'll not give you any written notes on dinoflagellates because let's start it from the next one. I'll explain what these do. Now, I think there was a, like I don't watch Hindi movies, but somebody told me actually what dinoflagellates do. Everybody knows what fireflies are. Yes? Fireflies which glow in the night. The Lucifer insect. Okay? So, fireflies glow in the night. Dinoflagellates have this property, some dinoflagellates, they are bioluminescent. Like, that means at the night time, they will make the sea look colorful. They will glow in the night. Okay? It will look like tiny stars are present in the sea water. It looks very beautiful. So somebody told me that there is this actress called Shraddha Kapoor, and there was a song in which she was there, and she was singing and something, and I haven't seen that, but I showed the pictures to the boys about dinoflagellate, and they told me that there is a song in which Shraddha Kapoor is singing, and in that song video, she is sitting in a sea water which is glowing brightly. So I told them that it is definitely dinoflagellate. So I'm not sure, you might have seen it. I haven't seen that song yet. I'm not a fan for Hindi movies. Okay, and no girlfriend either. So no point in seeing such things. Anyway, what I'm trying to tell you is, there are dinoflagellates which can turn phosphate into phosphorus, and phosphorus can cause phosphorescence or glowing in the night phenomenon. Okay, so these are examples of dinoflagellates. Besides this, dinoflagellates are responsible for red tide. Suddenly sea water becomes red in color. Then they are responsible for food poisoning by eating poisonous shellfish, because they produce some poisonous toxins called saxitoxins. And so there are many things. So that we'll cover in the next class. So I will also show you the images of diatoms. I will show you the images of dinoflagellates and so on. So that will help you in remembering all these things. Okay. So dinoflagellates is going to take a bit of time. about 2-3 minutes are not sufficient for it. Okay. So thank you, dear students. I'll meet you in the next class, and let's see if we can do some questions based on this. Okay. So thank you for your time, and we'll try to solve as many questions as you can from the module. And please feel free to ask me, to contact me if you're having any doubts. So I've covered the maximum, like it's more than the syllabus actually, but not like what's given in your boards. So it's definitely more than the board syllabus. Plus, please don't be worried, because I'm covering everything that is asked in the NEET examination. Okay. So we'll start in the next class from dinoflagellates. Okay. Thank you. Thank you for your time. You can go now.